So our next speaker is Dr. Eran Perison from, actually from the Sackler Faculty of Medicine, not from Life Science. Please move him back. We like him. And he's going to talk about molecular communication mechanism of motor neuron, survival and synapse maintenance in ALS. Can you talk now?
Yes. So, okay, thank you very much. Good morning, everyone. So, maybe a sentence regarding Miguel's talk. So, I think one of the main things is to keep things in balance in the cells. And if things are too much or too less, it can be toxic. So, over-autophagy can be toxic. So, I will show you today a way to study mechanism of ALS, a novel platform that we create in the lab. And we will suggest also a specific mechanism that may be responsible to some of the ALS patients. We think, I guess like many, that ALS is a syndrome of many different alterations and different mechanisms. And what I will show you today is maybe one such alteration. Okay, so as we heard before, there are many cytoskeleton alterations in genes that involve in ALS. And in order to put things in order and to emphasize maybe why it's important, we need to remember that neurons are highly polarized cells. They have a soma in the spinal cord or in the brain, and a very long extension called axon that can reach to the muscles in the periphery. Okay? So I don't know, I guess like many of you, I watch the late at night, the NBA players, and each time I saw LeBron James jumping to give a dunk, I was thinking about its long sciatic nerve and how accessible it is to difference in axonal transport process. So neurons are highly polarized cells with very long axons, and in order to survive and maintain normal function, they depend in this process that's called axonal transport. Movement of proteins and organelles like lysosomes or autophagosomes from the soma to the synapse and from the synapse to the soma. This axonal transport process, you can think of two major roles: to supply or clear factors and organelles, and the other to create a spatiotemporal signaling event that is essential for the neuronal health. So we were talking about difference in gene expression. Not less important is the localization of the gene and of the proteins and the factors. And maybe to be a little bit, I don't know, I would say or suggest that any neurodegenerative disease, including ALS, involve in alteration in localization and in the cytoskeleton. And there are evidence in that. So how does the axonal transport is conducted? Axonal transport is conducted by motor proteins, the kinesin and the dynein, that are walking along microtubules, which are like the railroads on the cells. These motor proteins using ATP energy in order to generate kinetic movement, and by that they can carry these different organelles and factors. When we are looking on an axon, we can see many of these organelles moving to the different sides, from the soma to the synapse, and from the synapse to the soma. And when there is alteration in these types of process, it leads to cell toxicity and death. So how this axonal transport process is regulated? You can think of four main levels of regulation. The first level is the microtubules tracks. So whether there are microtubules or not microtubules will influence, of course, on the ability to move stuff. So these microtubules stability can be regulated either by post-translation modification like acetylation and other, or by different proteins that can stabilize or destabilize the microtubules. For example, the famous tau from the Alzheimer, or plus end microtubules like the EB1, and many researchers in Tel Aviv, including Illana and other, are working on these pathways. Another level of regulations are the motor proteins themselves. Those motor proteins, the dynein and the kinases, can be phosphorylated, they can be activated, they can be recruited to the different cargo. Another level is adaptors and the ATP themselves, which is mainly supplies from the mitochondria. So altogether, if you can think, are actually the reason for most of a neurodegenerative disease. We know that there are neurodegenerative diseases because of mutation in the microtubules or the microtubules mechanism. We know that a mutation in dynein and kinases can lead to motor neurons disease. We know that different adapters like Crab, Huntington's, and other can lead to neurodegenerative disease. And we all know about the difference in mitochondria, localization, and dysfunction that can lead to alteration in ATP. So in our lab, we are studying molecular communication mechanism of motor neuron survival and synapse maintenance in ALS. And when I'm referring to molecular communication mechanism, I'm talking about communication within the neuron difference compartment, communication between the soma, the axon, and the synapse, but as well as communication between the motor neuron and its diverse extracellular environments, with initial focus on communication between the motor neurons and the muscle that create the neuromuscular junction. So in order to study this type of neurocommunication, we decided to create a novel platform and to grow both motor neurons and muscle cells in a microfluidic silicon chips. So basically we have a silicon chip where we can put on one side of this compartmental chamber motor neurons, you can see it here in green, and on the other side muscle cells. The motor neurons grow their axons towards the muscles to form a neuromuscular junction. And then we can study these types of mechanism and neurocommunication. So this is how it looks. You can see the green neurons passing through the chip grooves where they meet a muscle cell. We can track the growth of the neurons. You can see how the neurons grow to the muscles, and when it's reached the muscle, it starts to innervate and build the neuromuscular junction. We can do different genetic manipulation in order to study the growth and the innervation of the muscles. And we now comparing between different ALS models to control in order to study these types of mechanism. You can see here an example of an in vitro neuromuscular junction. You can see the green presynaptic side and the red postsynaptic side of the neurons. This is a confocal imaging showing how this neuromuscular junction is found in vitro in these microfluidic chips. So we also wanted to show first that these neuromuscular junctions are indeed functional. And for that, we first looked on muscle contractions. We thought that when the muscles are innervated, they are contracting. You can see here how the muscle contract when they are innervate. And when we applied TTX, a toxin that basically inhibits the action potential at the presynaptic side, we saw inhibition in the muscle contraction, suggesting that indeed this NMJ, in vitro NMJ structure, are functional. We further used different types of functional assays, including FM dyes, uptake and release. We did also some calcium imaging, showing correlation between elevation in the calcium in the neurons and in the muscle that again can be inhibited by adding the TTX at the presynaptic side. Using this system, we can also track axon degeneration mechanism. For example, when we apply oxidative stress at the distal side, at the neuromuscular junction side, we can see how this process leads to toxicity and to axon degeneration. very interestingly, when we applied same stress to the soma side, to the cell body side, we didn't saw any toxic effects, suggesting that the neuromuscular junction is much more vulnerable to stress and to toxicity, while the soma side has a much better ability to deal with stress and so to summarize this part, I'll show you that we develop a microfluidic platform with motor neuron cell bodies on one side and muscle cells on the other, that this platform exhibits a powerful system to study communication mechanism between motor neurons and the muscle, that the platform allows precise control, monitoring, and manipulation of subcellular microdomain, and that this platform provides a simplified way to study mechanism of neuromuscular junction formation, maintenance, and disruption. So basically now we have a platform that we can really study basic mechanism of neurodegeneration. And I think this is very important because I think, or my belief is that we do not have yet a treatment or drug for ALS is because we do not know what leading to disease. And if we will have this knowledge, then it will be much easier to develop a specific drug and treatment. So with this part, I will now move to show you an idea of maybe a possible mechanism for toxicity. And we heard about ALS, but what I would like to emphasize is that one of the early things that occur in ALS is the disruption of the neuromuscular junction that can lead to a dying back mechanism. This is thought to be one of the early things in ALS. And together with Oded Bar from the Hebrew University, we suggested a model that maybe in ALS there is elevation in semaphorin molecules that usually are expressed during development and are guidance molecules that have a repulsive role, and basically this elevation in semaphorin from secretion from the muscles can lead to neuromuscular junction disruption, axon degeneration, and neuron death. So to test this idea, we first show, and I will not go on all the data, but we show that different semaphorins are elevated in muscles, at a different model of muscles, both in the proteins and in the RNA level. We also show that the receptors of those semaphorins are elevated in the neuron side. So there is on one side more secretion of semaphorin from the muscles. On the other hand, there is increase in the receptors for those semaphorins in the neuron side. And we did it using real-time PCR and Westerns and staining and so on. It doesn't matter the details. Well, it's hard to see because of the light, but looking also on the neuromuscular junction itself, we can see okay, never mind. We see also that in the neuromuscular junction themselves, there is increase in both in the semaphorin and in the receptors for semaphorin. Okay, maybe we do need to turn out the light a little bit. Okay. Oh, okay. Now you can see it better. So we thought that there is an increase in the semaphorin, increase in the receptor. What does it mean? So when we treated in our chambers the green motor neurons with semaphorin, so for example treating with control, we see that the axons are growth and very happy. However, when we treated it with semaphorin, you can see that after some time you see very strong degradation of the axons. Further, when we looked specifically on ALS axons, so you can see here 3 green axons, 1 red that represent overexpression of the mutant SOD, and I will play the movie and you will see that these axons that expressing the SOD is degraded much faster than the axons that doesn't express the ALS gene. So you can see now degradation of the motor neurons expressing SOD, and only later the degradation of the control. So this suggests that there is elevation of semaphorin in ALS models, and that this elevation in the semaphorin secreted from the muscle together with elevation of the receptors expressing in ALS models can lead to toxicity. But we also asked why the semaphorins and the receptors are elevating in ALS. And as we heard before, there are many different mutations in ALS related to RNA metabolism. So our hypothesis was that maybe alteration in the RNA metabolism can lead to increase in the semaphorin and semaphorin receptors. And indeed, we did a screen, deep-seq screen, comparing two different ALS models, the TDP and the SOD1, comparing between the total RNA in the axons and the soma. And I will not go it very quickly, but between the microRNAs that can regulate the levels of the proteins, we found some that are regulated both the semaphorins and the semaphorin receptors. So altogether, I'll show you today that elevation in semaphorin and in semaphorin receptors, because of alteration in microRNA metabolism in ALS models, can be toxic and maybe can explain some of the cases of ALS. Of course, the next stage will be maybe to design specific drugs that can target the semaphorin and the receptors, and by that to rescue this toxicity. So, okay, so by that I will summarize, I will acknowledge my team. I have really good group of people that working on different aspects of the ALS disease. I will thank you, my collaborators and funding agencies, and if there is time for a question, I will be very happy to answer on questions. So thank you.
So thank you very much, Eran, and I must congratulate you also on a great progress since last time we heard you. And this is open for questions, please. Yes, please.
This is an increase in semaphorin in the mouse model of ALS?
Yeah. So the increase in semaphorin is both in vitro and in vivo. And yeah, interestingly, we see only 30% of the neuromuscular junction in vivo increasing the receptor semaphorin, suggesting that only part of the neuromuscular junction are subject to this mechanism. We are now trying to understand whether these NMJ are the most susceptible in ALS, if there are these fast fatigue NMJ, those neurons that are more active and so on.
Yes, Miguel.
So, Eran, do you think the ALS is triggered by the periphery, the muscle is telling the neurons to die, or which environment affects which?
Yeah, so I don't know. This is the question. I think that there are now a lot of evidence suggesting that the peripheral compartment is first to fall. But whether it is because alteration in the soma or local change in the synapse, this is something that needs to be...
But I think we need to take also the first talk that Vivian showed us, all the genes that are mutated, and see whether some of them are more neuronal specific, because the failure of the cytoskeleton that is coming up could be more neuronal, I think. So it's probably a crosstalk.
Well, I think for sure you need, I think that there are many evidence suggesting that it's alteration in the neurons and in the environment. So both of them together can lead to disease. So whether it's muscle and neuron, muscle with astrocyte and so on, it's, I guess it's...
And these miRNAs, microRNAs are turned up or turned down, are they known to be downregulated or upregulated by many different genes, or affect all the possibility of 20 genes that are being associated with
Yes, so what I would like to say now is that we found so we didn't find many microRNA, but those that we found alter both in the SOD and in the TDP models are downregulated, and those that are downregulated can, according to some bioinformatic analysis, can regulate both the neuropilin and the semaphorin receptors.
And this also goes with the work, the Weizmann of Eran Ornstein.
With Eran Ornstein, yeah. So Eran Ornstein have shown that if you knock down dicer and you see downregulation of the microRNA, you can see some ALS-like phenotype.
So last question.
Did you try to give those some protein by siRNA or maybe antibodies against the
No, but so actually there was a paper published, I don't know, this year, that this is what they did. So they use an antibody against semaphorin 3A, and they show some rescue effect. So yeah, but what we see is that it's only this, not only the semaphorin 3A, but there's also other semaphorins, and we also show why there are upregulation. So yeah, so it's done.
By the way, there is a new paper in Science with the C9 open reading frame 72 model for ALS, so this might be also interesting to look at.
So this is also something that maybe I would like to say, is that as Vivian said, although there are many different genes that were involved, including now the C9 ORF72, that now it's like the gene that everyone in ALS trying to understand, because it's still the phenotype of these mice is very mild. I mean, the mice are not dying and do not have ALS. So they have, it's true, all kind of phenotype, but they, unlike the SOD one, they don't really generate ALS-like disease.
Okay, thank you very much.